Welcome back to the second week of the online portion of this class. Hopefully you're settling into a pattern. The videos are working for you. If you need help or whatnot, contact me. Hopefully you had a chance to look at the assignment and that went pretty well. If not, gives you a way to target your studying or again to contact me if there are questions. By way of announcements, I did push the exam back until this coming Monday. So a week from today. I will have an online virtual study session, so I will give you more details about that, but that'll likely be Sunday evening or sometime on Sunday, so stay tuned for that. We're going to continue to work our way through Chapter 7, which is about cell signaling. And so I just wanted to kind of point out generally, right, we're talking a lot about action potentials and the electrical activity of the neurons and whatnot, and you might think, well, why are we doing this? Why are we talking about how cells communicate? And it's because a lot of the pathways that we're going to get into today underlie some really fundamental neuronal processes, things like long-term potentiation, which is required in how we learn, or some of these pathways underlie enzymatic synthesis pathways of neurotransmitters that we saw last time. So this may feel too general, but I promise that this content will find a home in this class as we move forward. So I think we left off, and I wanted to talk a little bit about the receptors that are targets of these ligands in neural transmission, ones that work not, what type of receptors do they activate, and then how does that pass on a signal? So these are four broad categories of receptors, and I think that it's important to know that the neuronal response is always going to be determined by the type of receptor. Right, so we can say that, I don't know, GABA mediates inhibitory neurotransmission, but that has to be through a GABA A receptor, right? And don't forget there's a metabotropic GABA B receptor, okay? So what are these four receptor subtypes that we can talk about, or the four receptor types we can talk about, I should say? The first are what we could call channel-linked. We've had a lot of experience with channel-linked receptors before. Right? In this case, the receptor and the signal transducer are put together. Right? So the protein that's on the cell surface, the receptor, transduces or passes on whatever message that ligand has directly. So the receptor is linked to the signal transducer. And what I mean by signal transducer, again, is just what's going to relay the message. And so, like I said, we've seen a lot of these because these are our ionotropic receptors. Right? Ones where ligand binding here causes this channel to, which is typically closed, right, to open up, form a pore, and now ions can get in. So this protein here in the membrane serves as both the receptor as well as the thing that responds to the ligand all in one. And so our result is ion flow across the membrane. We see a depolarization. The second type of receptor we can mention are what are called enzyme-linked receptors. In this case, the ligand often activates an enzyme directly. So whatever signaling molecule we have, whatever ligand we have, and so up here what we see is a common type of enzyme-linked receptor called a tyrosine kinase, an RTK, a receptor tyrosine kinase. We'll talk about them in more detail. But in this case, what happens is the ligand, the signal, binds to the receptor directly, right? But that doesn't open any ion channel that allows this enzyme to now do its catalytic job, right? So it is going to generate a product. It's going to cause speeding up of some chemical reaction from substrate to product. Okay, so we'll talk about maybe one specific example, right? This RTK, or receptor tyrosine kinase, in a little bit. The third type of receptor is the one that we're going to spend the most time on tonight. The G protein coupled receptor. So G protein. Coupled receptor. I've been abbreviating this for a while, GPCR. Okay. These are going to regulate all kinds of reactions within the cell indirectly. So the G protein coupled receptor isn't going to participate in any of the reactions that it regulates. So regulate intracellular pathways, reactions, however you want to think about this. I'll say reactions, but it's an indirectly sort of way. Indirect. How? Well, as you can see in this diagram here, it has a lot to do with this G protein that associates with the GPCR, right? So here's the GPCR, that's the receptor, and the G protein down here is going to be the thing that transduces the message, right? If we're looking, as we talked about in example A, what's transducing? In this case, it's the G protein itself that's going to transduce the message. So regulate intercellular reactions by or via G proteins. And again, we're going to get into much more depth. I think the way that we're going to attack this is work our way from general to specific, okay, but we want to be all-inclusive. So you know some examples of this already. We talked about beta-adrenergic receptors. GABA B receptors, receptors for the neurotransmitter dopamine. These are all G protein coupled receptors. I think something like 50% of drugs that target the nervous system target G protein coupled receptors. So this is going to be a huge focus for us, okay, as we go forward in this chapter. Lastly, we will mention and talk about some examples of what we think of as intracellular receptors. These receptors require the ligand to be cell permeant, okay? So in this case, there's nothing on the surface with respect to the receptor, right? And on these three other cases, the protein was bound to the surface. In this case, these signaling molecules are able to move, and typically through diffusion, right through the cell membrane. And the receptor is often in the cytoplasm, okay? So the recellular is within, right? Intracellular. So this requires cell permeant ligands. So cell permeable ligands. So a lot of the ligands that we've talked about in the last chapter with respect to neurotransmitters, anything that's an amino acid, right, typically won't cross, or a derivation of an amino acid won't cross the cell membrane. Certainly a peptide won't cross the cell membrane. So these tend to be more specialized, not a large category of molecules. What they tend to do is target DNA and change transcription and thereby translation. So their changes aren't going to be nearly as immediate as, say, the channel-linked receptors, right, which we think of as fast activating, right? Change happens quickly. Intracellular receptors, out of all the four of these, change is going to happen the slowest because they're going to influence the entire genetic program of the cell, right? So maybe they modulate, regulate, kind of the same thing. DNA expression. right? Modulate, like modify. They're going to tinker with DNA and how it gets expressed. And so typically what happens is once this receptor is activated, it's going to go off and have some effect on the DNA. Okay, so 4 types of receptors. Let's look at those G protein coupled receptors in some more detail. So the way we're going to look at the cell signaling and with respect to G protein coupled receptors and a couple of other receptors too, is we're going to not look at any one signaling pathway. We're going to talk about general effects of each of them. Okay, so there are 2 classes of G protein receptors. You can see an example of each on the right. Not class 6, excuse me, that's classes. The first and probably the more complicated are what are called heterotrimeric G protein receptors. Okay, and so the G protein, the G protein itself is made of 3 different subunits. Alpha, beta, and gamma. And alpha typically associates with a GDP or GTP. This is where the G in G protein comes from. Associates with GDP, right? You know ADP and ATP. GDP and GTP are similar, right? So if you think about the structure of ATP, it's adenosine triphosphate, right? Adenosine is a nucleotide. So go back to what you know about DNA, right? There are 4 nucleotides, A's and T's and C's and G's, right? Adenine, which is a lot like, it's almost, I think, with one functional group difference, adenine and adenosine are the same, right? G's, guanosine, right? That's this. Okay, so it's a nucleotide with phosphate groups attached to it. Okay, and so if we look here at the G protein, the alpha subunit, this one here, is associated with the GDP. Okay, guanosine diphosphate. Now, what happens is when the ligand, the chemical signaling molecule here, right, neurotransmitter or whatnot, binds to the receptor, the G protein associates with it. Okay, so under normal circumstances, the G protein isn't necessarily nearby, right? So this whole complex of alpha, beta, gamma, right, with the alpha portion having GDP associated, is somewhere not near the G protein coupled receptor, okay? But ligand binding changes that. Ligand binding brings it to the receptor. Oops. So ligand binding allows the G protein to associate with the G protein coupled receptor. Okay, so now this whole thing is together, right? And that's what's shown right here in the picture. Everything put together. So when the 2 associate together, something happens to the G protein, and we'll pick it up here next.